Morning. Apologies, I have the flu and therefore I couldn't present in person in Cairns. So I really hope this recording will work. George and I will present some of the work we have been doing to predict the dispersal of coral larvae in the context of coral larval restoration, which is led by Professor Peter Harrison and Dr. Christopher Doropoulos in the Moving Corals subprogram within RRAP. I am a postdoc researcher at Southern Cross Uni, and George is a senior experimental scientist at CSIRO. Moving corals interventions are all field-based that take place during major spawning events, when buoyant coral gametes rise up to the surface and form slick, which we target and collect, and then transfer into rearing ponds. In those rearing ponds, we let larvae develop until reaching competency. Once larvae are competent, we then transfer them using various methods onto the reef to kickstart their early recovery. So as our operation occurs on the reef, the intervention are subjected to local environmental conditions, and the one that has the most influence for us is the current. So as you know, ocean waters are constantly in the move. They, especially at the surface, we call them surface current. They occur in the top 50 to 100 meters of the water column. So this current are usually set by the wind that drags the surface of the water into one direction and then creates flows. But there are also interacting factors playing here, such as the Earth Coriolis effect, the shape of the seafloor, tides, river inflows, wave, and local weather conditions. And from studies measuring currents on coral reef, we know that, for example, current speed can vary up to 15-folds among coral reef habitats. So luckily, there has been lots of effort over many decades into modeling hydrodynamic environment in various habitats and at various scales. And we are making use of this accumulated knowledge to test fine-scale biophysical model and its ability to predict coral larval dispersal during restoration intervention. So here you can see the different models that we have trialed and tested over the past few years. So the first one was in 21 and 22 called the Liz model, which has a donut shape and a domain of 8 by 8 kilometers with a fine resolution close to the reef that increases as you move offshore. This model has a nested regular grid of about 50 meters to allow for particle tracking. And it was integrated with CONI. Then in 2003, we upgraded to an unstructured grid that basically looks like a honeycomb, as you can see on that photo. So the domain is approximately the same size, 8 by 8 kilometers, and we tested two different models, one around Lizard and one around Eyrie Reef. With this modeling workflow, the particle tracking component is embedded within the modeling suite as form as plugins that were developed by Jack Beardsley from CSIRO. That basically allows automated processing, which become really useful when we are in the field. So these models are three-dimensional. They operate at a 10 to 12 minute time steps. They do have 30 meters resolution near reefs, and they provide a forecast hydrodynamic condition and associated particle dispersal output up to 6 days ahead. So having this forecasted condition is very useful during fieldwork to predict spawn slicks convergence zone. So here you can see on the left, we have forecasted hydrodynamic condition during spawning nights last year. And on the right, we have the relative reproductive output of reefs around Lizard. Here we used coral cover, reef area, and the relationship between cover and spawn produced on reef. We then integrate this information into the particle tracking component of the model to model spawn slicks. So we get a forecast of particle concentration during predicted spawning nights, and we can identify what we call convergence zones of coral spawn slicks, which is where high concentration of coral spawn will likely form. So this helps us make decisions on where to go what we call hunting for spawn at night, for example, but also where to deploy spawn catchers a few days before a likely spawning event. So here you can see the forecast of particle concentration around lizards throughout one of the spawning nights at 10 p.m. midnight, 2 and 4 a.m. the next day. The model also assists us in predicting dispersal during delivery of coral larvae on reef. So when larvae in the larval pools are competent to settle, we then transfer them onto damaged reef. And we often have only a 5 days time window to do that. And during these 5 days, hydrodynamic condition can be very variable because of the current, the wind, the tide, and so on. So we use the model to forecast the dispersal of larvae during 12 to 24 hours following delivery, hourly from 6 a.m. to 6 p.m. for 5 to 6 days. And the goals of this simulation are first to identify location, day, and time of the day, where local retention is highest to maximize local settlement of a damaged reef, and second, to predict the footprint of larval releases. This is an example of some of the findings from the modeling exercise we did. So we dispersed a set number of particles at 25 reefs in the southwest region of Lizard Island during delivery condition in 21 and 22. And we calculated the residence time of particle within 1 hectare of the delivery location. So overall, we found that out of these 25 sites, 20 sites had one or more occurrence of particle residence times over 5 hours. And some sites show residence time of 12 hours quite frequently. So these sites therefore make ideal candidates for larval releases from a hydrodynamic point of view, of course. Through this exercise, we also found that slack current condition, which is the time periods on the reef where the current is quite weak and could lead to high retention of larvae, are completely unrelated to the timing of high and low tide, and therefore tide is a very poor indicator on when to release larvae on reef if we want to maximize their retention. So overall, during free releases of larvae on reef, the timing of larval delivery is key, and the model can assist us in predicting this.
So as Maria's outlined in the previous section here, in terms of upscaling the larval restoration, the hydrodynamic models have been able to provide insight into one of the key questions of larval reseeding, which is where do larvae disperse following release? So from the tens of millions of larvae released onto damaged reefs during free releases, how many settle in the immediate area, and what are the spillover effects into adjacent habitats? And the next logical question from this is where do the larvae settle following release? So, and this is important in terms of converting the number of larvae into expected densities in terms of a production rate, but ultimately we're looking to quantify the spatial footprint of larval restoration. To quantify this, we developed Coral Seed, which is a spatially explicit probabilistic model of larval settlement that leverages these high-resolution dispersal models. So Coral Seed is an individual-based model, allows upscaling from small-scale simulations of thousands of larvae to large-scale simulation modeling of releases of tens of millions of larvae. Coral Seed is spatially explicit in terms of incorporating these larval particle tracks, but also interactions with habitat maps. So where are larvae passing through space and time, and are they passing over habitat that could be suitable for coral settlement? So it's also adopts a Bayesian framework, so it's probabilistic in terms of determining settlement outcomes, and importantly, it's parameterized on experimental and field data generated under the RRAP components. So to give an example as to how Coral Seed works, here's a trial run from Porphyry Reef at Lizard Island, and this is based on simulations we ran back in 2022 during the mackerel spawning in December. So in contrast to 2022, here's what Porphyry Reef looked like a few weeks ago, March 19th in 2024, showing more than 90% of the corals were bleached and the onset of mortality in these corals following prolonged thermal stress. And this is a reality that faces many of the reefs on the GBR today. And upscaling restoration efforts need large-scale responses in order to be able to mitigate some of this damage moving forwards. So this little box in the top left of the map here is our release site on our restoration hectare, and we're simulating 1,000 release particles from this release box. So these are the larval dispersal paths. This rainbow color shows the dispersal from 0 to 12 hours following release. So when larvae settle following release is a challenging question, and it's key to understanding larval reseeding approaches. So to better understand this, we conducted a series of experiments, both at CSIM and during coral spawning at Lizard Island, to parameterize this time to settlement model. And this is a Bayesian modeling approach that determines the probability of each larvae becoming competent, and then maps the competency to these simulated dispersal pathways to determine when and where larvae are in space and time when they become competent to settle. So the spatial component of the model is then enhanced by using the island coral atlas layers to determine the seascape configurations of different habitats for reef back reef and reef slope. And then based upon larval activity experiments, each of these different habitats has a different probability of settlement for coral larvae based upon larval preferences. So we then take these habitat maps and turn them into a mosaic of settlement probability, which allows us to generate seascape mosaics of settlement probability, where in this map, the darker colors represent a higher probability of settlement on the reef. So combining probability here, we can then determine the potential spatial locations of larvae that settled on the reef, and then the proportion of larvae that were diluted and lost to off-reef dispersal. So focusing in here on the fine-scale settlement patterns on the south of Porphyry Island, you can see these grey dispersal tracks and then the final settlement locations of the larvae in the dots below. And one of the benefits of these spatially explicit models is that we can then quantify not only the total numbers of larvae settled, but also the underlying spatial patterns such as settlement density, but also the spatial footprint of settlement, which allows us to evaluate the impact and the scale of these reseeding programs for the first time. So in summary, the coral sea model aims to provide insight into key knowledge gaps in larval restoration. Where do larvae settle following release onto reefs? And what is the spatial footprint from larval reseeding projects? What can be attributed to settlement in the area we're looking to restore? And what are the benefits from spillovers into adjacent habitats? What are the likely densities of settled corals we can expect when restoring damaged reefs? And then what densities of adult colonies are produced from reseeding, and where are they located? So an additional thing we've been looking at doing with coral seed is how do we vary the initial model parameters, and can we really test the ecological limits of larval reseeding? So to what extent can larval reseeding scale towards large-scale restoration? How do we take these small-scale efforts and look to restore damaged reefs, such as entire reef flanks, kilometers in length? And importantly, we've been looking to see whether additional interventions can improve on investment and return as part of the larval reseeding. So Marin's already introduced an example of this in terms of optimizing releases to coincide with slack tide periods. But we've also been looking to see how we can enhance larval retention through the use of bubble curtains to entrap larvae, or acting as barriers to restrict dispersal, which in turn enhances settlement, or how incubating larvae with inducers such as coralline algae prior to release may reduce competency times and accelerate the settlement onto damaged substrates. And finally, we've been exploring how time-delayed releases can improve settlement densities by releasing larvae when they're competent to settle, and that has an immediate impact on settlement following release, as opposed to delayed dispersal. So in summary, Coral Seed is written in R and is open source and will be available for anyone to use. And importantly, the model is flexible in terms of parameterization and works with a diverse range of dispersal models. The code is available on GitHub, and a manuscript on the underlying theory and the applications of the Coral Seed model is currently in draft form. And with that, on behalf of Marin and I, we'd like to thank CSRO and SCU, as well as the RRAP for funding model development and fieldwork as part of the Moving Corals program. And special acknowledgements to Lizard Island Research Station and the staff at CSIMS for accommodating are experimental and field-based research.
I had one. I thought so in Marine's presentation, so what she mentioned that was a mismatch between the tide and where the larvae ended up. Was that the case?
The mismatch that Marine referred to was a mismatch. As field ecologists, we often think that at the high or the low tide periods, the slack tide periods is actually when you get low current velocities. But because of the dynamic nature of coral reefs, that would probably work in the open ocean, but that doesn't work on a coral reef consistently. So some parts of the reef might match up with the slack tidal periods, but other parts don't match up as well. Yeah, Clotilde, maybe you can add to that or correct anything that I did incorrectly. Yep.
Okay, any further questions? Okay, thank you, Chris.
Oh,